Gamete intrafallopian transfer, also known as gift, is used for infertile women who are ovulating but have blocked oviducts or fallopian tube. Gamete intrafallopian transfer uses multiple eggs collected from the ovaries. The eggs are placed into a thin flexible tube, or known as catheter, along with the sperm to be used. The gametes, both eggs and sperm, are then injected into the fallopian tube using a surgical procedure called laparoscopy.
Most babies are conceived by a sperm fertilizing an egg and the fetus growing inside the mother's uterus. But sometimes this is not possible. One or both parents might have fertility problems. For lots of different reasons, some parents are unable to have children. In fact, there are 5 million children worldwide born to parents who either couldn't or had difficulty producing babies. One way of helping with infertility problems is through IVF, and has been in use since 1978. IVF stands for in vitro fertilization. This sounds complicated. By the end of this video, you will know all about how IVF works. IVF can be used when natural fertilization inside the body cannot occur. This is often due to the reproductive parts of the mother or the father not working properly. In IVF, an egg is removed from a woman. Outside the body, sperm is introduced to the egg and fertilizes it. The fertilized egg develops to form an embryo, and the embryo is then returned to the woman's uterus, where the fetus then develops in the uterus as normal. We will look at this process in more detail later in the video. The wonderful thing about IVF is that it can overcome a multitude of fertility problems. If a woman has a problem with her eggs, then donor eggs can be used. Or if the male sperm are the problem, then donor sperm can be used. Or maybe the sperm and the eggs are both healthy and compatible, but the woman's uterus is inhospitable. Then a surrogate mother can be used, but with the mother's eggs and the father's sperm. So let's have a look at the IVF process in a little more detail. The first step is called downregulation. Drugs are used to turn off a woman's ovaries by suppressing the production of the hormones FSH and LH. We learned about FSH and LH in the menstrual cycle video, which you may want to watch to remember how they work. In brief, FSH and LH are the hormones that trigger egg development and ovulation during a woman's monthly cycle. Turning FSH and LH off suppresses a woman's natural menstrual cycle, making the stages of treatment more effective. This happens for about 2 weeks. The next step is to boost egg supply. This time, lots of follicle-stimulating hormone, FSH, is injected for about 10 to 12 days. FSH increases the number of eggs your ovaries produce. This means that there will be more eggs available to be collected and fertilized in the treatment. More eggs increases the chances of success. Once enough eggs have been produced, another hormone called HCG is injected. This causes the eggs to mature so that they are ready to be released from the ovaries. The woman is sedated and a very small pipette is used to remove the eggs from the woman. The eggs are outside the body and ready for fertilization. With the eggs removed, the woman is now given progesterone hormones to prepare the lining of the uterus to receive the embryo. Now up steps the man. Sperm are collected into a petri dish where they are processed and the healthiest ones are kept. Healthy sperm have a higher chance of survival and are most likely to fertilize the eggs. The egg and sperm are then mixed together in a petri dish and left to fertilize for about 20 hours. If the fertilization happens, the embryos are kept in a warm place set at human body temperature to replicate the mother's uterus. They stay here for up to 6 days to ensure they are starting to develop into embryos. As I said earlier, the woman is given progesterone to make sure her uterus is ready to receive the embryo. The best 2 or 3 embryos are then transferred back into the woman's uterus by using a long plastic tube called a catheter. This procedure is actually really simple. 2 or 3 are chosen to increase the chance of survival. IVF is complete and the natural embryo development continues inside the woman's uterus. Watch part 2 to discover some of the advantages and disadvantages of IVF treatment.
If you liked the video, give it a thumbs up and don't forget to subscribe. Comment below if you have any questions. Why not check out our FewSchool app as well? Until next time.
Zygote intrafallopian transfer or ZIFT involve oocyte retrieval by the transvaginal ultrasound guided aspiration, followed by culture and insemination of the oocyte in laboratory within 24 hours. The fertilized eggs are transferred by laparoscopic technique into the end of waiting fallopian to the tube. ZIF different from GIF, and the fertilization take place outside the body, allowing healthcare provider to be certain that fertilization has occurred before the growing structure is reintroduced. As in a GIF, a woman must have a functioning fallopian tube for technique to be successful, because the zygote are implanted into embryonic end of the tube rather than into the uterus. This technique allows for genetic analysis the same as IVF. The technique invited fertilization or IVF removes several eggs from the ovary to fertilization to the laboratory. After a few days, 1 or 2 in this fertilized egg return into the uterus and they form in a plant to become a pregnancy. Women having a IVF are special reproductive hormones to encourage several eggs develop into the ovaries. Final matination is the egg cell is the use of administration for nurse's hormone. Within 36 hours later, fluid contained the egg is drawn into the ovary with the needle. This usually performed at the light sedation in short outpatient procedure in the use of ultrasound to check the proceeding. And the egg collect from the ovaries, mixed from the sample of the male sperm, and already contracted the egg sperm are left in incubator and settled in 36 degrees in 24 hours so that fertilization can take places. During this time, only one of many sperm cells is prenatinated outer layer of the egg cell and form fertilization. The following fertilization separated form into the embryo. After 2 to 3 days, the healthy embryo will compromise around the egg cell to the transfer into the uterus form implant to become a pregnancy.
Surrogate embryo transfer. Surrogate embryo transfer is an assisted reproductive technique for a woman who does not ovulate. The process involves use of an oocyte that has been donated by a friend, relative, or provided by an anonymous donor.
The menstrual cycles of the donor and recipient are synchronized by administration of gonadotropic hormones at the time of ovulation. The donor's ovum is removed by a transvaginal ultrasound-guided procedure. The oocyte is then fertilized in the laboratory by the recipient woman's male partner's sperm or donor's sperm and placed in the recipient woman's uterus by embryonic transfer.
An embryo can be transferred back into the uterus. After 2 to 6 days of culturing in an incubator, a very thin, flexible plastic catheter is inserted through your cervix. And with the help of a small syringe, the embryo is injected into the uterus. If the embryo implants in the uterine wall, it will release a chemical signaling the pregnancy.
Preimplantation genetic diagnosis, the individual retrieval of oocytes and their fertilization under laboratory conditions have led to close infection and recognition of differences in sperm and oocyte. The preimplantation genetic diagnosis is an early form of prenatal diagnosis in which embryos created in vitro are analyzed for well-defined genetic defects. Preimplantation is a technique that enables couples with a particular inherited condition in their family to avoid passing it on to their children. In preimplantation genetic diagnosis, we have the techniques are used mainly in two broad indication groups. The first groups are individuals at high risk of having a child with a genetic disease. The second groups are those being treated with in vitro fertilization who might have a low genetic risk but whose embryos are screened for chromosomes aneuploidized to enhance their chance of ongoing pregnancy. In preimplantation genetic diagnosis, we have IVF and GIFT. The in vitro fertilization or IVF, a medical procedure whereby an egg is fertilized by a sperm in a test tube or elsewhere outside the body. The GIFT are modified version of in vitro fertilization. After the oocytes are fertilized in IVF and GIFT procedure, the DNA of both sperm and oocyte can be examined for specific genetic abnormalities or specific genes such as Down syndromes or hemophilia. So we have 8 typical treatment cycle for preimplantation genetic diagnosis. Number 1 is stimulation of the ovaries. In order to create embryos for genetic testing, the ovaries have to be artificially stimulated using hormones to produce several eggs at the same time. The number 2 is collection of egg from the ovaries. At the appropriate time, the eggs are removed during a surgical procedure known as egg retrieval. Once the eggs are removed, they are inspected to determine which are mature and normal appearing. The number 3 is insemination or injections of sperm. Fertilization during PGD will be done either in IVF or in ICSI. In IVF, sperm and eggs are placed together in a culture dish to allow fertilization to occur. While in ICSI, a single sperm is injected in the center of an egg. This technique is used for condition caused by a single faulty gene. The number 4 is fertilization. The morning after insemination or injection of the sperm, the embryologist carefully examines each egg to see if fertilization has occurred. Number 5 is embryo biopsy. The eggs that were successfully fertilized are grown in the laboratory for 5 to 6 days. When the embryo forms, that is called a blastocyst of about 100 to 150 cells. Number 6. Embryo testing. The cells are tested to see if the embryo from which they were removed contains the abnormality that cause the genetic conditions. Number 7. Embryo transfer. Only those embryos that do not have the specific genetic condition that was tested for will be transplanted into the woman's uterus. And the number 8 is the pregnancy test. 12 days after the embryo transfer, the woman is given a pregnancy test. A positive pregnancy test means that an embryo has implanted. A surrogate mother is a woman who agrees to carry a pregnancy to term for a subfertile couple. The surrogate may provide the ova and be impregnated by the man's sperm. In other instances, the ova and sperm both may be donated by the subfertile couple or donor ova and sperm. Surrogate mothers are often friends or family members who assume the role out of friendship or compassion, or they can be referred to the couple through an agency or attorney and receive monetary reimbursement for their expenses. The subfertile couple can enjoy the pregnancy as they watch it progress in the surrogate. Surrogacy process occurs in 4 steps. 1. Finding surrogate mother. 2. Signing surrogacy contracts. 3. Fertilization and pregnancy. And last, claiming parentage after childbirth.
There are many would-be parents today who are unable to conceive and/or carry a child naturally. Unlike many years ago, however, today there are some very viable options available to help these would-be parents have the child that they so much want and deserve. The process of surrogacy entails the use of one woman's uterus to implant and carry the embryo of a child, as well as to deliver the baby for another individual or couple. The woman who carries the child is referred to as a surrogate mother. Here's how the surrogacy process works. Most intended people start their journey by filling out the surrogate mother application. This application asks about your lifestyle, pregnancy, medical and work history, which acts as a basis to match you with appropriate intended parents. Once your application is accepted, you will receive a call from a surrogacy coordinator who will gather more information about you and your family. You will then be briefed about the programs and options available, the detailed process of matching with a surrogate, the legal process, insurance issues, financial implications, and basic medical information. Once everything is in order and the selection of surrogate is done, introductions between the intended parents and the surrogate are facilitated. When the joy of pregnancy is achieved, you and your surrogate are prepared to welcome your child home. Child-free living is an alternative lifestyle available to both fertile and subfertile couples. For many subfertile couples have been through the recourse and frustrations of subfertility testing and unsuccessful treatment regimens. Child-free living may emerge as the option they finally wish to pursue. A couple in the midst of fertility testing may begin to re-examine their motives for pursuing pregnancy, and may decide that pregnancy and parenting are not worth the emotional or financial cost of future treatment. They may decide that the additional stress of going through an adoption is not for them, or they may simply decide that children are not for them to complete their family unit. For these couples, child-free living is a positive choice. Child-free living can be fulfilling as having children because it allows a couple more time to help other people and can contribute to society through personal accomplishments. It has advantages for a couple in that it allows time for both members to pursue careers. They can travel more or have more time to pursue hobbies or continue their education. If couples still wishes to include children in their lives, in some way, many opportunities are available to do this through family connections, through volunteer organizations, or through local schools and town recreational programs.